seminar series, and today we're going to get more into the evolution side of this, which I think is really exciting. So I believe Dr. Pinto is going to give a little bit of introduction to his background, but he is from Iowa. I am from Nebraska, so Midwest is representing today. And I think we may have first met in Chicago, also in the Midwest. So a lot of solid stuff coming out of there. But I am really excited. So Dr. Brendan Pinto joined the lab about 2 years ago, almost exactly in October.
Exactly, yeah.
Like through some sheer dumb luck, decided that he would like to come to the desert. Maybe we have some interesting lizards here. And since then has worked on a variety of projects, some very methodological, but I think today is going to talk to you a lot about organismal projects. So thank you so much for coming and talking.
Thanks, Melissa. So today I'm going to tell you about the sprightly little Sphaerodactyl. This is my dissertation work, which also came out of the Midwest, so we're still doing it. Let's see. So yeah, I did my PhD in Milwaukee, and I'll get to that in a minute. And then I've interlaced this talk with some more recent work that Melissa and I have been doing on sort of moving the field forward as a nascent. I doubt you guys have heard a lot of talks on reptile genomics, because there's not that much work that's been done on reptile genomics to date. We're trying to change that. So before I get into it, I've never published a single author paper in my life. I have a lot of people to thank. The people bolded here are the people that have collaborated on the work that I'm going to present today, including obviously Melissa, my PhD advisor, Tony Gamble, and then Dr. Nielsen, who's actually here today visiting, is up here. And most of the gecko photos you're going to see are his. And instead of plastering his name all over my talk, every time you see gecko photos and you see this in the corner, it means they're all his. Okay, so I'm not a photographer. I have him to thank for that sort of stuff. So it's great to have him here, and we're going to jump on in. So a little bit about me. From Iowa. Went to a small college you've never heard of. I did my PhD in Milwaukee under Tony Gamble, studying geckos, specifically Sphaerodactylus, or the Sphaerodactyls. Published a lot of cool work describing species, resource development, that sort of stuff. That's sort of been a hobby of my scientific career, is generating things for other people to use, mostly because I can't do it all myself, nor would I want to try. And mostly focused on geckos. For the past 2 years, I've been here at ASU, also a member of the AGA, on the executive council as an early career researcher, and I'm an associate editor for them now. So that's sort of a little rough back and forth there on who I am, what I'm doing, and why I'm talking to you right now about lizards. So the work I've done in lizards can sort of be broken down into a few different pieces. So I started at phylogenetics, population level, species delimitation stuff. And then I moved on to resource development, transcriptomics, specifically of the eye, was a starting project for me. And then I moved on to, once you start doing omics, you just keep moving up. That got to the genomics, which is the work I'm going to be talking about today, the sprite little Sphaerodactyls genomes. And then more recently, Squamate genomics is more of a broad field, and why it's exciting and new and cool, and more people should be involved. So today I'm just going to talk about what's on the right. So here's an outline sort of for today's talk. We already took care of the introduction. You don't need to hear any more about me specifically. So I need to give you a little background on genome evolution. So we're going to start with just planting a seed in your brain. What is synteny? Why it's important. Sex chromosome evolution, as we're the sex chromosome lab. This is fundamental in what we do and study and what we're interested in. And then why would we study this in squamates? And then I'm going to tell you about the Sphaerodactyls and squamate genomics more broadly. So synteny, we all know we've got these 4 base pairs. They're in long strings, they're wound up into chromosomes, and those chromosomes evolve, right? So we know that sequence-wise, a lot of conservation across vertebrates, but the order and linkage of those sequences varies drastically. So if we take for instance, the Z chromosome in chicken, and we look at where the Z chromosome of chicken is evolutionarily, we see it jumps around. It's broken up in mammals. It's maintained in linkage groups on autosomes and other reptiles. And so I just want to plant this seed in your brain that not only do sequences evolve, but the way in which sequences are organized also evolves. Okay, so plant that. We'll get back to it later. Done with the synteny thing, sex chromosome evolution. So animals need some way, sexually reproducing animals need some way to bin some animals as male, some as female. This is done through two common mechanisms, either genetic, such as with sex chromosomes, where either the male or the female possesses a sex-limited chromosome that initiates the primary sex-determining cascade, or some environmental, such as temperature dependence at some critical point in development that influences whether you turn out to be a male and a female. Again, we've got great photos from Stu on this slide. So, and across life, there's a vast diversity of how animals determine sex and whether or not animals are even, or organisms are even sexually reproducing. But again, mammals and birds sort of doing one thing. They got this single color here, and then across other organisms, you see a lot more variability. And so what is this variability and how do we account for it? So it's odd, it's sort of counterintuitive. Sex determination is really conserved and it's super variable. And what does that mean? It means that this top link in the chain here, the primary determiner of sex, can vary drastically across organisms. But at least in vertebrates, this downstream stuff is relatively very conserved. And so we focus less on this in the lab because it's very conserved, but this is very interesting because it's variable, it's replicated across organisms, and that's super interesting to us. So where do sex chromosomes come from? Why are we interested in them? So sex chromosomes evolve from an ordinary pair of autosomes. One of the two acquires a sex-determining allele, something that trumps whatever was there before. That autosome or nascent sex chromosome becomes the Y or the W. Over time, recombination is suppressed, and the sex-limited chromosome begins to degenerate. So this is what's happened in mammals and non-ratite birds. But actually, most vertebrate species possess sex chromosomes that look like this. You can't see the difference between them under a light microscope, which has made them historically very hard to study, seeing as how most of what we know about sex chromosomes was, for 100 years or so, was devised from looking under a microscope. So this is where we're going to be. We're going to be up here in this realm with the most vertebrate species. Now that we've sort of niched the sex chromosome thing, why squamates? Why reptiles are the best? Now here we go. There we are. We've got the piece of the pie that's all sorts of different colors. X, Y, Z, W, temperature, other stuff, XO, XXY, all that stuff is in there. And again, this paper's old. So old is relative, less than 10 years, but still out of date because the field has moved so rapidly recently. Why squamates? This is a pie chart of all amniote species, okay? Just numbers. We have A, B, C, and D. I'm going to orient you to what a slice of this pie looks like by telling you that this little slice here is turtles and crocodilians. Next, I'm going to ask for a show of hands who thinks that mammals on this pie chart is the number A, or the letter A. My bad. The number A is also, great. You're wrong. Who thinks mammals are B? Got some smart people in the room. Those are mammals. Yes. There's a lot of mammals, but comparatively in amniotes, they're the small chunk of the pie. And so now we have one more question. A or C, are there more birds? Or are there less birds than squamates? Can I get a show of hands for A, or 1? There are more birds than squamates. They got 1-ish, okay. And who says 2? There's everyone else? All right, you guys are good. All right, squamates are the most numerous group of amniotes by a reasonably large margin. And we're still describing them in such a way where the number keeps going up. Whereas the taxonomy of other groups tends to be a little bit more stable. And so within this chunk of the pie, we have a vast diversity of really cool traits: sex determination, reproductive mode, prey defense, locomotion. As you know, snakes have no legs. Legs have been lost a bunch of times throughout lizards. So it's just a lot of cool traits. And today we're going to focus on just one of them, and that's sex determination. And so if you look at a phylogeny of just the squamate species, this is, again, a nearly 10-year-old paper showing a diversity of sex determining modes. So here we have gray is temperature dependence, blue is XY, and orange is ZW. And if you looked at an updated version of this, which nobody's generated quite yet, it would look even more chaotic than it does in this photo. And so for today, the purpose of today's talk, we're going to go up to this group here, this clade that seems to be doing something very different than most. Those are the geckos. Specifically, we're going to focus on this 3 taxon statement, which happens to be the Sphaerodactylidae. And so moving on to our sex chromosomes in geckos. This is a prune phylogeny of the 13 genera that make up the family Sphaerodactylidae. This is all the information we pretty much know about the family and their sex determination modes. So we know that the genus Aristologer has a conserved ZW system across the genus. We know that Euleptes has 1 species that's known to be an XY. Gonatodes has 1 species that's known to be an XY. And Sphaerodactylus has 2 species that are known to be XY. And that's it. We know nothing about linkage, evolution, conservation. We know nothing else. This is pretty much where we started when I started my PhD. And this figure is actually from the first paper I was a co-author. So enter the sprightly Sphaerodactylus. So sphero is sphere or ball, dactyls is toe or finger. So these are ball-toed geckos. They have really cool adhesive digits where there's a ball at the very tip. That's the adhesive apparatus in these animals. They're neotropical. There's over 100 species. They're really cool sexually dimorphic animals. These 2 here are male. These are sexually dimorphic species. These are sexually monomorphic. But again, they're diverse, they're small, they're cute. They got everything going for them. And if you look at them from the top, they can move their eyes independently and just like look at 2 people at once. And it's just really cool. So I have to give you a little background on the sprightly Sphaerodactyl. This comes from a quote on, so this is a paper by a Major Chapman Grant. This is the last surviving grandson of Ulysses S. Grant. He was a herpetologist. He did some seminal work in Puerto Rico for Sphaerodactyls. And I just got to read some excerpts of his one of his first papers on the subject. And the Sphaerodactyl is little known to many collectors. It's hard to find, harder to catch, and still harder to classify. That is, unless you enjoy working in the open and turning over innumerable rocks or scraping seemingly acres of dead leaves. Hard to catch unless you can make up your mind to grab a fraction of a second sooner than the uncover prize makes up his mind to dart. And he is a creative creature in quick decision and action. Hard to classify unless you have a large series to work with, thus avoiding the confusion of growth phases and sexual dichromaticism. I know of no genus that more amply repays finding, catching, and classifying than the sprightly little Sphaerodactyl. And my PhD advisor and I got on a kick when we were publishing a paper a while ago, and we said, the sprightly little Sphaerodactyl sounds like an amazing children's book title. And so that was the cartoon on my opening slide, and I will end with that as well. But we commissioned that art inspired by this this very poetic intro paragraph to this paper. This is a prune phylogeny of Sphaerodactylus. So this clade here is all from the Puerto Rican bank, and Sphaerodactylus notatus here at the top is our outgroup for the talk. It's from Florida and Cuba, basically, but it's very distantly related. You'll notice the break here that it's about 30 million years diverged from everybody else. So when I started my PhD in 2015, this was all that we knew about this group. So we knew that Sphaerodactylus nicholsi had an XY system shown here on this gel. And we know that Sphaerodactylus inigoi has an XY system also shown here on this gel. And that's it. That's all we got. Starting over from scratch here. And so some methods, some data we generated. So we did RADseq. RADseq is sort of a staple of my PhD lab. It uses a data set of multiple males and multiple females, and basically identifies alleles that are present in one sex and not the other. Okay. And then we generate a reference genome, which I'll get into more later. And then we did a variety of different ways to parse apart, basically read mapping and identifying sex chromosomes. So RADseq is pretty simple, like I just said. You cut up the genome at some specific site. You analyze it in some way, which you can filter out or things that are in one sex and not the other. All the species I'm going to talk about today have an XY system. In this mode, you'll get markers that exist in males that aren't present in females. Oh, sorry. And then you can validate this on a gel by designing PCR primers, and it becomes very clear that the locus is clearly present in one sex and not the other. And so when we did this, we see a consistent pattern of XY systems, like I said, in all these species. We have a lot of male-specific markers, but very few female-specific markers. And so, again, we can plot that information on a phylogeny. And, if we were doing this work 5 years ago, we would have to have stopped here, right? We don't have genomes. We can't really do much else besides that. But fortunately, we do have genomes, so we can do more. And that's where my PhD went. So once you know that everything has an XY, for instance, in this clade, for instance, there's actually multiple different ways that you can get that pattern, right? So it could all be the same XY, an ancient conserved XY system that all Sphaerodactylus share. It's certainly possible. And we'd find that they have a conserved linkage group that is the XY in all these species. You could also get, our outgroup has like a different XY, and our focal group from Puerto Rico maybe has the same XY. And this is sort of a pseudo-conserved model. Or we can have an every clade for itself model, where maybe sister taxa have one system and then another clade has a different system. Again, you can imagine a bunch of different scenarios where you have some amount of conservation or no conservation between species. And so to get at this question, we generated a reference genome for Sphaerodactylus townsendii, one of our focal taxa. We did whole genome resequencing for a male and a female. A few of these, like I mentioned, had RADseq data, and then we had a little bit of RNA-seq for the last two that we were able to use for these analyses. So one of the things I'm going to get to later is genomics and squamates more generally, but one can imagine a situation where this is the distribution of the number of species across amniotes. A pie chart mirroring this should, maybe be what you'd expect for available reference genomes. Maybe the numbers change, but you'd imagine the proportions might stay the same, right? The proportions differ very much, and this is not filtered for if they sequence the same goat 20 times or something, but mammals have the most assemblies. This is just NCBI data, have the most assemblies. Birds are numerous. Again, a very stark contrast to what we see in the actual diversity of these organisms, and there's reasons for that, some of which I'm not going to get into, but this is a problem, and it caused us to need to generate our own data, right? And we needed to do a reference genome, and not only is that serving us, but serving the whole field, because, well, we need help, put it that way. What this allows us to do, generally speaking, is just assign chromosomes to sort of their evolutionary counterparts across a diversity of organisms. And doing that in squamates has already yielded some pretty cool insight. So we can use this reference genome to map all of our RADseq data. and identify the linkage group, that's the sex chromosome in these animals. And what we'll see is that in Sphaerodactylus townsendii, where the reference genome comes from, and its sister species, Sphaerodactylus nichollsi, see this sharp, obvious peak on linkage group 3. Inigoi has this really interesting dual whole chromosome thing going on. And this is, again, a pattern that's consistent with the XY system we saw earlier. And then we see something fundamentally different in our outgroup, Sphaerodactylus notatus. And so just zooming in, I need to convince you that this is a real signal in Sphaerodactylus townsendii, and then I trust that you'll believe me for the rest of the species, okay? So 87% of our RAD-seq markers that we identified that are male-specific mapped right to this region that has high divergence between males and females. And we can even put that on a gel after generating PCR primers and confirm that indeed there are regions in this genome that are male-specific. And so now I'm just going to blast you with data for a few seconds here. And we're going to look at conservation across these things. So species that have observed or patterns on linkage group 3 that's consistent with it being a sex chromosome are Sphaerodactylus townsendii, nichollsi, Clauberi. This is one of the ones we had whole genome resequencing for, and then Inigoi. So in these 3, we see this one sort of chunk here of linkage group 3 that's very different between males and females. Inigoi, like I said, we have this really long stretch that it's on the same linkage group, but it looks different than these 3, obviously. And then this is an autosomal pattern in our other 2 species as we look down the phylogeny. On linkage group 1, we see 2 species that have sex chromosome consistent patterns on linkage group 1. Again, in Inigoi, we see this really long stretch of divergence between males and females. And then in notatus, it's just this one spot. Interestingly enough, that one spot is in a different position. It doesn't overlap with the spot here in Inigoi, although it's the same linkage group. And then this linkage group seems autosomal in the other taxa that I just mentioned. And then probably important to mention, we don't know what the heck's going on here. No signal, any chromosome, certainly nothing consistent with all the other species we looked at. So this is going to and is an open question. But we can plot that back onto the phylogeny I showed you earlier. And so we can assign linkage group 1 to our outgroup, linkage group 3 to our in-group, linkage group 1 and 3 to Sphaerodactylus inigoi. And this could happen through a variety of mechanisms, but I'm going to tell you that it's actually a fusion, an XY fusion, or an X autosome fusion in Sphaerodactylus inigoi. It's why it's got a really big chromosome. So it looks like they're 2 separate linkage groups, but it's actually one in that species. And so that begs the question, and this is where I will sort of conclude this part of the talk, is linkage group 3 the ancestral sex chromosome in this clade? And we don't know. That would be cool to look at, but a dissertation is only so long. You got to stop somewhere. So this is where I stopped. Another thing, again, the differences between conclusions 1, where we knew that everything was an XY, and conclusions 2, where we know the linkage groups and can formulate some evolutionary hypotheses. The difference is the reference genome. The reference genome also allows us to do cool stuff, like look at the actual gene content of the sex chromosome. And so if you actually look in Sphaerodactylus townsendii in the region that we know is the sex chromosome, we identify a very strong candidate as a primary sex determining gene, the thing that's extraordinarily variable across taxa. And in this species, it's AMHR2, or anti-Müllerian hormone receptor 2, which is a pretty common sex determining gene across vertebrates. Similarly to AMH, which is a pretty common sex determining gene across vertebrates. And these are all, this table illustrates all of the putative sex determining genes that we know of in squamates. Yes, 4 of 11,600 and whatever number I showed you earlier. So we got some work to do, but we're getting there. And this is all driven by the rise of genomics being available and useful and cheap for squamates. And so last, I want to just touch on some community resource stuff for Squamate genomics. There we go. So this, as of July of 22, this is all the chromosome level genomes that were available for squamates. There's just 24 of them. Actually, first we published that in January, so that's sort of cheating. But so there's 23 squamate genomes as of last year, published at the chromosome level. Again, these are annotated, useful genomes that anyone can sort of go online and grab and ask questions. And before this, and I'll show you here in a moment, nope, yep, nope, yep, nope. There we go. Freeze. I'll show you in a moment that most of these have been sequenced in like the past few years. Exponential increase in genome, in useful, high quality genome assemblies. Like I said, there aren't very many. Again, if you just remove the numbers, these proportions are out of whack, which also presents an interesting opportunity in squamates in that we're a young field and we have an opportunity to do things right, do things better than mammals and birds. I mean, the organisms themselves are better, right? So so we can do things a little different than have been done in the past, potentially. So it's important to know that even of those 100-some genomes that I showed you a little bit ago, they're not evenly distributed across the phylogeny, as you may have guessed. There's a lot of snake genomes, particularly driven by interest in venom evolution. But most families still have zero references. Until just a month ago, there was no chameleon genome, for instance, not one published at all. And now there's 3. That's great. But there's still work to be done. So here's a sort of a time course of squamate genomes as they've been generated by year. On the top here, this is total genomes in gray, chromosome level in black. And on the bottom here, it's total genomes again, the same gray as the top one, but whether or not it's annotated on bottom. And what I can tell you is that quality is improving exponentially in conjunction with the numbers, which is great because not only has sequencing gotten cheaper, it's also gotten better. So this is really awesome. And mind you, these data cut off in July of last year, so that's why it, seems like it's a little stunted on the last year. But annotation remains a challenge. All right, so there's certainly realms in which we can help each other. Right, good annotation groups can help help groups who aren't good at it. Right, we can establish sort of a network of people who can help each other make use of these genomes in a better way. And then one thing that I will specifically point out is that not only is this recent explosion really awesome for the community, it's also entirely biased as where these genomes are being generated from. So 75% of these assemblies come from the global north, 21% from China specifically, and then the rest of the global south generated 3 of these assemblies. And that's just not a pattern that really works for everyone, right? And so there's ways in which we can potentially try to alleviate some of this. And the answer might be, and what we're going to try to do, is start a Squamate Genomics Consortium. So it's just a way to get people to talk to each other. And easier said than done. But again, this is sort of just starting this month, but we're trying to extend collaborations across the globe, saving people time and money. For instance, there are 2 Komodo dragon genomes available. Both of them are pretty bad, but they have complementary data sets where if the people generating them would have talked to each other before publishing, the final product would have been much better. And there's dozens of cases like that where we can fix this by simply just providing a way for people to communicate with each other. And what this will do, we hope, in general, is empower genomics people who are interested in genomics in the global South, particularly. So we can sort of try to fix a little bit of this stuff. Because we're so young, we haven't gotten so ingrained in our ways of how we do things. Maybe we can start now before it becomes too late. And last future directions, sort of for this work, Melissa and I are excited to start working on some more Arizona herps. So we're going to do some sex chromosome and genomics work for native species of night lizards. There are 3 species here in Arizona. It's 1 species of gecko we're excited to work on. The Gila monster, of course, we're going to dive in a little deeper than what we've done already. And alligator lizards, a relative of the Gila monster. There's 1 species that lives here, and so, well, why not, right? And we're doing this in collaboration with Anthony Barley and Levi Gray at ASU West Campus and the Arizona Genomics Institute down at the University of Arizona. So more exciting work to come, more exciting lizard talks to be had. And with that, I will take questions.
There is morphological differences between.
Yes. So there are morphological differences between males and females, and then that's what you're starting for comparing males and females, or are you also trying to determine the sex and then see if it's a sex, determine if it's XY or temperature dependent? That's determining the sex of those lizards. Does that make sense?
Yeah. So they are morphologically distinct. They have an interesting scale pattern in the lower regions that are specific to males. And then generally speaking, the males have bright heads. Not in all the species, of course. But since we know that this area of the phylogeny doesn't have a ton of temperature dependence, we sort of just rule that out.
Okay.
Usually, and not always, once you transition to a sex chromosome system, you don't generally go back to a temperature dependent system. I mean, it happens, and we've seen it, and we've found some interesting transition taxa across squamates, but it seems pretty rare.
Okay, thank you.
It's an interesting talk, Brendan. Thank you. My question is there any evolutionary consequences of this sex determination and maybe reproductive effort in these lizards? It grabbed my attention that geckos, they only lay 1 or 2 eggs. So
Yeah.
What's different in the genetic configuration, maybe these chromosomes in geckos compared to iguanids that can make 20, 50 eggs. So is there any thoughts on that?
It's unknown why. But what I would tell you is that yes, geckos, an interesting aspect of geckos specifically is they have a fixed clutch size of 2. There's a couple exceptions, one being Sphaerodactylus. They only have 1. And that's because we think they're physically too small to have 2 eggs. The females would explode. You should see a gravid female. They already look like they're balloons. Right. So they also lay one egg a month. So they're just constantly churning offspring out. And they don't really have a breeding season. They breed year round because they're in the Neotropics. There's not a ton of variability there. And so they're just constantly laying eggs. Yeah. As to why that is, I don't know. Couldn't tell you. I know it's true, but I don't know.
Thanks for that talk. That was really interesting. So you were just saying that when they go to like a sex chromosome determination, that they usually don't see the reversal, right? Which suggests that somehow it's better. I mean, that's at least what to me, it would suggest like it's better. Otherwise, there would be some kind of evolutionary pressure to go back. So is that true? Like, is it better? And do you have any idea like how, in what sense? Like, is it just you get more even sex ratios? I know when we talked last week, you said like it's pretty even sex ratios, even with temperature. So I'm just wondering like if there's any kind of hypotheses about like what determination is better, quote unquote. And that probably depends a lot on environment, I would imagine.
Yeah, I don't think either is better. But what I will say is that, especially in something like a gecko, where the ancestral state is, in a lot of clades, is temperature dependence, in order to get to a genetic mechanism, you have to have something that trumps temperature. And so you start sort of stacking on top of each other. This trumps that, trumps that, trumps that developmentally. And if the first thing you get trumps temperature, it's unlikely that temperature is going to come back and trump the genetic mechanism, if that makes sense. It's possible. And again, we do see it in some taxa, but it's sort of difficult to wrap at least my head around how that would work.
I guess if they would like lose that particular chromosome, and if there's more evolutionary pressure to have the environmental temperature, the sex determination, then you would see reversal, right?
Potentially. Again, it's a lot of chicken and egg situation. Like even if it was beneficial, what are the odds that it appears and fixes, right? Like it's like there's multiple layers to that actually sweeping through a population. And in organisms like this that specifically Sphaerodactylus that weather hurricanes and stuff, drift may or may not play a lot of role in what exists and what doesn't. So I would say most of this is probably drift related. I don't think that there's a ton of selection against genetic or temperature dependence, but that's just sort of my opinion.
On the slide where you had the linkage groups 1 and 3 with the chart, and there was the one species that was undetermined. If you had to speculate, what do you think is most likely going on there?
Oh God. One, I will say that is the species that we have the least data for, and the data itself is the most suspect. I'm not sure I believe anything. We've done RADseq on them before, and it's the most chaotic thing I've ever seen in my like FST across everything is just like a huge spike. There's something is wrong with those data, so it needs to be just done from scratch. I can even begin to speculate.
Do you think they're classified correctly?
Yes. So we've also done the systemics on this group. In fact, we published a paper called the Sphaerodactylus, where we did the systematics on the Puerto Rican clades and Virgin Islands clades, and it seems to be sort of well resolved. But what I will say is that the two species that the one with the two peaks across the two chromosomes and Macrolapis, the one that we don't know anything about, they hybridize sometimes, and those hybrids appear to be sterile. And so it would not surprise me if it was a totally different, just a totally different system, and there's a lot of conflict between them. So again, I hesitate to speculate with no data.
So you said that these squamates, they don't have a reduced genome of the sex chromosome as we have, like in the X chromosome or the Y, the Y chromosome. It's not stripped down. It has features of an autosome.
Yeah, some species do.
Oh, some people do. Is there some particular advantage or disadvantage to having one of the sex chromosomes be all stripped down? Like our Y chromosome is so, barren, except for the set. Is there some advantage or disadvantage, to one or the other?
I would say the advantage would go to not having a reduced sex-limited chromosome, if you want to, classify it that way. The problem is once recombination is suppressed, it's a feedback loop. It's called Muller's ratchet, where recombination is the main mechanism as which chromosomes repair damage, and without that, things can go sort of wild. And that's what happened in mammals, for instance. Once you stop being able to repair things, again, things just go off the rails. And
sorry, the theory for how we it's all sorry. What we know about this is based on so few taxa that are boring, right? Mammals and birds.
Sorry, mammal and bird people.
Hey,
Truth hurts. Jokes.
It's very, as a mammal person, I have to admit that. But I wonder how we still know very little about rates of recombination in males and females across species, and so there might be some interplay. Generally, males have lower rates of recombination, which exacerbates the effect of if a sex-limited chromosome is in the species that has a lower rate of recombination, then you could get some weird things. And I don't know, do you know in the lizards any of these?
There's
They have
no
similar rates?
recombination map published for, or no sex-specific recombination map published for any squamate.
Okay, yeah, I would have been surprised, but I had to ask.
Yeah.
Why do we have lower recombination? Because they're different.
Oh, why
chromosomes?
are they? Oh, males just have no recombination in many species, either no or very limited, low rates of recombination. So when we look at recombination maps, they're usually sex averaged. And then, yeah,
So
no,
the
it's-
males are just like true to type of the parents, whatever the parents' genome was. Is that it? Like
without cross, I mean, there are crossovers, but the rates tend to be much lower,
like
or absent.
in hymenoptera,
Yeah.
the males are just basically it's like a sperm it's like a flying sperm. Basically, the males just completely reflect the mother's genome.
So a little different than that, but a conceptual.
Yeah. But it does vary. Like in the last species of frogs, recombination only occurs at the very tips of the chromosomes. So once you have a new sex chromosome system, most of the chromosome is non-recombining, and it very quickly degenerates across the length of it, except for at the very tip. So again, and so that could be a way, and again, that just rapidly degenerates. I'm glad we had questions because I ended early, so this is great. Yeah.
That was a cool talk. Thank you. I was curious about how you assembled the reference genome.
Oh, yeah.
Like what type of reads did you use? Was it like a combination of long and short? And then what way did you use to scaffold it chromosome level?
Yeah. So that's a great question. And full disclosure, most of the technology is out of date already. But so we started with a linked read assembled genome from 10x Genomics. We assembled it using Supernova, and then I gap filled using about a 3x coverage of Oxford nanopore reads. That was a fun little, I bought a MinION and thought it was cool project. That's grad school for you, by the way. And then we scaffolded it using Hi-C from an ARIMA genomics kit. And that work was all funded by the AGA, so.
Hey, so just to build off of that question, because I was kind of wondering it in my head too, I'm looking at generating some reference genomes for my dissertation, and I was just curious, how did you come up with coverage to start? Because it seems like with so minimal genomic tools available for this group of organisms, I'm kind of looking at something similar, and I'm looking at a range of like, okay, do I want to be extra conservative with my coverage or start low, high? And I'm also deciding was curious about long and short reads as well. So
Yeah. so mind you, this work was started 5-ish years ago. 10x genomics was the thing. It was the next, it lasted for a hot minute, but at the moment it was next level. And now I would never go back to a short read genome, not in a million years. I would go, and we will be going with PacBio hi-fi sequencing as to get sort of your primary assembly. If that, for whatever reason, isn't good enough, you can add Oxford Nanopore to make it sort of T to T. But I've never seen a bad assembly from PacBio hi-fi reads. I mean, weird taxa that do weird things, but it's at least some vertebrates. I've never seen a bad assembly from those. And then for lizards, at least, we generally assume the average lizard genome is 2 gigs. And so we do all of our math on that. Our genome is a little smaller than that, is about 1.8. So we sequence to 2 gigs. And at the time, with short reads, it was more the better. With PacBio and stuff, depending on what you need, 30x is fine. So.
Thank you.
Yeah.
You mentioned for birds and mammals that a lot of the genomic work was maybe not done the best, and for squamates, you have the opportunity to do it better than before. Is a lot of that just because technology has advanced, or maybe also strategy is different now that we've done it a lot of times for other organisms? Can you speak on that?
Yeah. So a lot of past work has been focused on essentially a small group of people leading an effort. And so everything sort of tapers to some hierarchy of some sort. So you'll see consortiums like the Vertebrate Genome Project, where you have to basically convince somebody to work with you to, again, you still have to pay. So it's not like somebody's going to do it for you, but again, you have to convince them that this is a valuable use of their time, et cetera, and then they sort of assist you after you pay them. That's not a super equitable way to do things. And in Squamate genome specifically, we actually did a quick analysis of like, how are they being funded? Because they're certainly not being funded by the NIH. Mammal genomes are NIH, bird genomes for somehow are NIH, but lizards are not. And so you'll see a lot of, there's a little bit of NSF, a little bit of USDA, but most of it's funded off like personal funds, startup, that sort of stuff. And again, it's tough. We need a grassroots effort where everybody can talk to each other, and not only can people like collaborate and work on each other, but know if somebody else is already doing it. Because this is valuable money, especially for someone in the global South who, you know, for us, $2,300 for a reference genome is sort of, that's a good deal. You know, the less you have, the more insurmountable that $2,300 becomes. And the way that costs drop, we see opportunities for, you know, better collaboration, more connectivity, more networking, less, more efficient uses of money across the board.
So for the consortium you were proposing or hoping to start, you hope to have it operate differently than the other consortiums for vertebrates, for example?
Yeah, I hope to mostly get everybody in a room and get them talking to each other, and, you know, stay out of it. You know, they don't need me to help. People are really good at self-organizing if you let them. I think that's how I feel, and I think that, again, if the network is there, if the infrastructure is there, whether it's like a, I don't know, Slack channel or whatever, people will start talking to each other as long as the methods or the mechanism is there. And right now, you know, I'm trying to connect people, and my network is this, you know, and I'm trying to get. It's hard to get people from outside of my network, because again, my network is like, it's a circular thing, and everybody is sort of has their own little, and again, I'm just trying to get everyone together, and it's a lot harder than it sounds. So
Good luck.
thanks.
So there was one question in the Slack, which I will take though, but I'll say it was, in humans, do they only recombine at the tips? Also, that's only true for the sex chromosomes. It's not true for the autosomes. The autosomes recombine across their length, and there are differences between males and females about where recombinations preferentially occurs, but only the sex chromosomes. So that was the answer on this one.
So I don't necessarily expect you to have an answer here, but
7.
that's wrong. Well, so I often think about like some of the things I learned about turtles a long time ago, and how there are species that actually have both like, you know, genetic sex determination and temperature sex determination. So basically, are you aware of any squamates where there's variation in sex chromosome systems, like within a species?
Yeah.
Okay.
So actually, the bearded dragon is one of these things where it's a genetic ZW mechanism, but temperature can override it in certain cases. And I think it's a high temperature. Yeah, you can get a ZZ female at high temperatures. I think is so it's a, you know, it's genetically or genotypically a male ZZ, but phenotypically a female. And again, that is hypothesized to be some transitionary phase where, again, temperature has the ability to then come back and override whatever the mechanism is. But we still don't even have a fundamental grasp on what exactly is determining sex in a temperature dependent system. It could be some sort of a signaling cascade, or it could be the temperature is actually affecting some protein conformation that is then doing something fundamentally different. And nobody has figured that out yet. So,
okay. So going further, do you think it would be a hard barrier between populations to have two different genetic sex determination systems?
yes, for the most part, I would say generally yes.
Okay.
I'm gonna leave it vague because
Yeah.
every evolution is much more creative than we give it credit for. So,
Okay. Yeah, because you mentioned that there were 2 sparrows that create hybrids that are sterile.
yeah,
And that. Okay.
yeah. So sex chromosomes are intimately involved in speciation. The 2 rules of speciation are Haldane's rule and large X.
Right.
Okay, Haldane's rule is that if 2 things hybridize and one of the sexes is going to be sterile, it's going to be whichever one is the heterogametic. So in mammals, for instance, you know, you get a mule, it's sterile. And that's replicated in birds, for instance, even though they have a ZW, all those sort of things. And then large X is just that the X chromosome evolves faster than autosomes because it has 3/4 of the effective population size as an autosome. And those things definitely affect speciation. Not a ton of work because most of this work has been done in mammals and birds.
Right.
Not a lot of to different sex chromosome systems.
Well, yeah, that's what's
hybridizing.
interesting about sparrows specifically, right? Is that like they've diversified pretty well, and that's way more variation than I would have expected in that small group of sparrows that
Yeah.
you work on.
More than us too. Tony kept telling me you're not going to find anything. Over and over again, he said this.
Super cool.
Yeah. And actually, the 2 species, the one where we didn't know, and the one that has the really big sex chromosome, I was able to hybridize them in a lab to an F2, and it was a weird looking sort of. It was a female, and it was phenotypically male-ish. It was just this very strange looking animal. And again, I think it's because we're dealing with some weird genetic conflict things where signaling pathways are trying to do opposite things. And so again, there's something to unpack there. I just am not the person to rear these animals by myself and do it. So.
Time for one more question. I'm attending remotely. Oh.
Yes.
Can you hear me?
His question just made me wonder, do you ever see parthenogenesis in these animals?
No. But here in Arizona, Levi's working on some of the parthenogenic whiptails and stuff like that. So there are other groups that are much better for that work. Cool. All right. Thank you guys so much.